of you that are here. I just want to introduce Dr. Johnson. He is one of the IR attending physicians here at MUSC. So today, as you all know, he's giving a lecture on device development and generally the field of device development in interventional radiology, but also maybe some personal things he's been working on as well. So without further ado, I'll let Dr. Johnson go ahead and take the floor.
Okay, let me just share my screen here. Can you guys see my screen okay?
Yes.
Yes.
All right, so we're going to talk a little bit about device development, particularly as it relates to interventional radiology. And yeah, we are going to talk about my personal experience with device development. So as a kind of outline of my talk, I want to talk a little bit about history. I like history, so I always talk about history whenever I give a talk, and a little bit about the pathway to develop a device, especially as it pertains to interventionalists, and then sort of my device, which is, that's it. That's what it looks like. All right, so history. When we talk about interventional radiology relative to a lot of the other specialties that you think about, this is actually a really, really young specialty. Anybody know who this guy is? The guy poking himself in the arm? Nobody? This is Werner Forsberg, and he's actually he's a surgeon, and this is in the 1940s. And at the time, people believed that if you put a catheter into the heart, that you would cause vascular spasm and the patient would die. And so to prove that that was wrong, Werner Forsberg, who was a surgical resident at the time, actually catheterized himself, and that's the x-ray of the picture that he took of himself. And he did actually lose his license to practice medicines, but he won the Nobel Prize. So I guess you can't totally be upset about that. And wrote a book called Experimenting on Myself. But really, IR was born in the 1950s, about a decade later. And this was with the work of Seldinger, who actually came up with the concept, okay, I got a blood vessel, I put a needle in the blood vessel, I see blood, I put a wire down the needle, I take the needle off, and I put a catheter in. And that's why we always talk about Seldinger technique. And this was sort of a really innovative way to actually get in without cutting big holes in people, which is what they did before this. The first angiogram was actually performed by a urologist back in the 1920s. However, he did it specifically by doing a big cut down onto the femoral artery and, like, you know, putting a catheter, sewing a catheter into the femoral artery. I think he actually occluded the leg on that patient too, but completely aside from that, this is kind of the innovation that led to all of our specialty. And, you know, this guy, Crazy Charlie, which is what everybody called him back in the day, Charles Daughter, he was actually famous for this. He invented all kinds of things. This is Laura Shaw, who was a diabetic, refused to get her toes cut off when she came in with a necrotic foot. And so he did angioplasty of her leg with the first guide wire, which he was using, which was the speedometer cable from a VW Bug, by the way, in case you're wondering what he used as a guide wire. And so this is all brand new when you look at this. This is 1963. Before this, there really wasn't a specialty, and it wasn't named until 1969. So this is a specialty that has grown up sort of in whole over just the last 60 years or so. And we started doing oncology at about the same time. These are spinal mets that people were embolizing back in the 1960s. We didn't even have cine imaging at the time. So these were cut films where they took a picture after they injected contrast, and they were embolizing these spinal mets, and they were embolizing them at the time. We didn't have anything fancy, so they were using things like ball bearings and macerated pig meat. They actually had a blender in the IR suites. They were putting macerated pig meat into some of these tumors, but some of this started to show promise, and that's kind of where all this developed. And these are some of the procedures performed in 1967. Image-guided in utero fetus infusion sounds terrifying to me. But aside from that, the rest of these we're still doing. And actually, we do a lot more. And in fact, IR has twice as many procedure codes as any other specialty in medicine. And so that's kind of where this whole concept of innovation comes from. It's part of our DNA. It's what we did. And if it was met with some resistance from other specialties, this is actually a requisition that Charles Daughter got the year after he fixed Laura Shaw's leg, which, as you can see, says angio, visualize, but do not try to fix. underline, 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 exclamation point, exclamation point, exclamation point. So apparently this this vascular surgeon was very clear on what it is he wanted. But we continue to innovate. In fact, Charles' daughter actually fixed one of the two disease vessels in this patient. The graft went down immediately after surgery, but the profunda stayed open, which is what he ballooned. And he did it with a dilator, not a balloon, because they didn't have balloons back then. But that spirit of innovation has sort of started with Charles' daughter and Seldinger and kind of went through to today. And part of the reason we did so well in terms of this innovation sort of culture and this innovation DNA that we have in IR is because we had strong ties to industry almost from the beginning. This is 1964, so the year after Charles' daughter did the first angioplasty, and Charles' daughter is the guy on the right, and the guy on the left is Cook, who founded Cook Medical. So as you can imagine, and these two worked extensively together. They're shaping catheters. That's why he's got a blowtorch on the table, just in case you were wondering what that is. But we sort of, from the very beginning, recognize that, in interventional radiology, completely different from other specialties, we are only as good as the devices that we have. And so you can't have the same sort of holier than thou attitude when you're talking to device manufacturers, because everything we do is determined by how good our devices are. And, there's stuff that the guys back in the 60s and 70s and 80s could do because they were super virtuosos with like garbage devices that we do very easily nowadays with devices because the devices have gotten so much better, and that just makes all of us better. And so this industry cooperation, this sort of culture of innovation has been with IR from the beginning. And even now, when other doctors come and talk to us, they're like, hey, I got a problem. What can you do to fix this? That is a conversation I've had hundreds of times since I've been in attending. And this is kind of where some of this comes out of. And, this is an awful lot of history for a talk about intervention, but I would say that it's important to remember that those who cannot remember the past are condemned to repeat it. So that's how I justify telling you a little bit about the history. And just to give you an idea of the speed at which IR has developed, and this is a mug that's sadly very true, Interventional Radiology Inventing Procedures for Other Specialties since 1973. Given that most of the things that you think about that are minimally invasive, all of them were invented by IR, if we're going to be real honest about it, including cardiac catheterization, cardiac angioplasty, all the uroscopic procedures. Those were all really invented by interventional radiologists. So a lot of them are done by other specialties now, but that's kind of where we come from and what we do. So our specialty, as I said, was named by Alexander Margulis in 1969. Actually, Charles' daughter hated the name, by the way. He thought we should be named something surgical, but Margulis recognized the centrality of imaging to what we do, and that's why he named it interventional radiology. He also wanted to keep it sort of united to radiology in general, and he was way more famous and way more influential than daughter, so he won. The Society of Interventional Radiology, which was the Society of Vascular Interventional Radiology originally, was founded in 1973. Subspecialty first accredited in 1991. The CAQ, Certificate of Added Qualification, which means you had to take a special board exam to actually be certified as an interventional radiologist, started in 1994, first exam in 95. It was approved as a primary specialty in 2012. And the first class of integrated residents graduated in 2021. So this year is the very first year that there are people that matched into interventional radiology who are out in practice. And I have 3 pictures up here on the screen. It's a shameless plug because those 3 people trained me. That's Ernie Ring, who invented TIPS, as well as all of the transhepatic procedures, portal procedures, biliary procedures, things like that. Him and Linderquist invented all those together. Jean LaBerge in the middle, who just retired last year, she's the one who actually did all the paperwork and got IR approved as a primary specialty. And Roy Gordon, I was his last fellow. He was one of the very first interventional radiologists in the country, and he designed a lot of the drainage catheters that we use in regular practice. They were all at UCSF. That's a shameless plug for the people who trained me, but there you go. And in addition to them, you have all of these people, many of whom are still practicing. Not all of them, but several of these people are still practicing. I know a ton of them, actually. These people created this sort of innovative environment where we developed devices over and over again, and things that weren't our procedures originally slowly migrated into our field of expertise. Okay, so that's enough about that. So the question is, how do you get from where you are to developing a device? Okay, so let's say you have a great idea for a new device. What do you do now? So it depends on where you are, but basically the process is very similar. If you work for industry and you invent a device, you don't get nothing. It belongs to them. Give up. You know what I mean? But many of you will end up in private practice, or you'll end up in academics. And so there's a pretty prescribed pathway for that no matter where you go. And I actually, for the next couple of pictures, I took pictures off the different websites from different universities just to make that clear. The first thing that you would do, no matter where you work, whether it's at the NIH, whether it's, in a military institution or at a university system, you would go to the Office of Technology Transfer, and it's always called the same thing. I think it's called the Office of Innovations here at MUSC. And basically what you would do is fill out one of these. And this is, as I said, I took one from different sites because they're all basically the same. And what this is what's called an invention disclosure. And this is really important to do before you talk to anybody, okay? Because within an institution, if you do an invention disclosure, then so it doesn't protect you in the way a patent does from other institutions stealing your ideas, but nobody within your institution is allowed to steal your idea if you have an invention disclosure that went to your technology transfer office. The thing that you really, most people don't recognize when they're thinking about inventing stuff is that if you invent something and you're an employee of an institution, they've all gotten wise to this, and it doesn't belong to you, it belongs to them. Because you invented it while you were working for them, presumably. And so you have to kind of have an idea of what it is you want to invent. You go through it, talk about what you think its potential market it could be and all that sort of thing. And then the university gets a chance to decide whether they think it's marketable or whether they think it's not. And if they think it's not, then you're still protected within the university, but they'll give you back the rights to it and you can develop yourself. If it is marketable, then they will start to engage and see what they can do to help you. So basically, you do your invention disclosure that protects you from anybody else in your institution stealing your idea from you. And then it also gives the university an opportunity to start working on what they can do to help you develop the device. And, it's a really good idea if you're really an invention-minded person to make sure that you look at what the rules are at your particular institution for device development before you go there, right? So as an anecdotal kind of story to do that, some institutions give you a ton of the IP back. So like at the University of Colorado, the rule was 25% of whatever the university gets for your idea goes to you, 25% goes to your lab, or 50% goes to you entirely, and then the other 50% goes to the university. And that's pretty good. That's about as good as it gets, to be honest with you. Some institutions are much less generous. So like at the National Institutes of Health, which have kind of the most interesting rules, basically you get every single penny of any invention you make up to the salary of a sitting U.S. senator, and after that, the university gets everything, or the institution, which is the NIH, gets everything. And so the most profitable patent, this is according to the director at the NIH, so I haven't seen the paperwork, but this is what one of the directors, the director of the Heart, Lung, and Blood Institute told me. The most profitable patent at the NIH is for birdseed, believe it or not. A guy in the National Institute of Allergy and Infectious Disease who did bird research noticed that birds don't have capsation sensors in their noses and mouths. So you can feed a bird capsation and it doesn't bother them at all, whereas mammals like you and me and squirrels, they do have capsation sensors. And so you can coat your bird seed with capsation and then the squirrels won't eat it, but the birds still will. Believe it or not, that patent is worth like $250 million a year or something. And the person who invented that patent gets $167,000 a year for, until they lose the patent rights or whatever. But that's a little depressing if you think about the fact that a patent that you came up with is worth that kind of money, and you get $167,000. So there you go. It's important if you're thinking that you're going to do this, that you should actually think about the rules at the institution where you're taking a job. One other trick that you can play is anytime you take a job anywhere, you were absolutely supposed to, when you walk in the door, which I did when I came to MUSC because I already had the idea. I went to the patent offices and said, hey, look, these are the ideas that I've already done before I got here. And so what that means is MUSC has no rights to them because they were created prior to my taking a job here. Does that make sense? And so that's one way to protect your IP, is not to develop it at the institution, but actually develop it before you go to a new institution. And so that's one way you can protect your rights, because anything you invented before you got to the university, they don't have a right to, except, and this is one caveat that's very careful that you should pay attention to, they have a right to any new permutations that you come up with after you get there. And so you really want to make sure that you're very clear about what the device rules are wherever you go, because this is super important in terms of protecting your idea. And so here's what you do. An inventor submits a confidential invention disclosure form, and this is the one from Tulane, just to give you an idea sort of how the thing goes. They take a look at it. They have their staff look at it, decide whether they think it's useful or not. In some places like Mayo, they have a specific fund set up to help you develop your design. So what they have engineers who work with them, and they have a fund that basically was funded by prior inventions that will actually put money into a device to develop it, to get it to a point that they can sell it and make money off of it. And you can make money off of it. And depending on what kind of device innovation they do, different institutions have well or less well-developed mechanisms for that. And so anyway, sometimes they'll bring it directly to market themselves. There are some investors who specifically sort of have relationships with institutions saying, look, I'll develop any device that you guys, I'll develop devices in your pipeline and we'll make money together. And then, that's kind of how it goes through. This is actually the MUSC development framework that you have here. And basically you do your disclosure, which is your, as we said, and then they do the assessment. They look at the market IP, they do some research, they do business planning, patent filing, all that kind of stuff, spin outs and licenses. And so this can take a long time or it can take a short time depending on what it is. But basically the way that this is sort of divided, it's, I think, in thirds at MUSC. I'm pretty sure it's in thirds, but anyway, so that's kind of how it all comes together. But you have to keep in mind, it's just really important for you to know your idea does not belong to you. So you could get yourself in trouble, and you could, and if you read your contract, when you sign a contract with a new hospital, you should always look at the, oh, no. MUSC is 50%. It's not, sorry, it's Mayo that's 33%. But MUSC is 50% of whatever the university gets, you get back too. It's really important to know what those numbers are. Some places it's really, really terrible. The terms at UCSF, you end up getting about 10% of whatever they make off your patent, which is awful. So you just make sure you're very careful about that sort of thing. So, and what the TTO, what do they think about when they're looking at your devices? They think, okay, is the idea original? Does it have commercial value if it is original? Is the idea protectable? Because, you can have a great idea. It can absolutely have commercial value, but if it's not something you can patent, that's a real problem, right? Because if you can't protect it, then other people with more money will actually end up making the money off it, not you. And what's the best path to development, whether it's develop inside the university, whether it's development with an industry partner, whether it's venture capital, all those sorts of things. Those are all things that you have to think about when you're deciding to develop a device. Okay, so you got a great idea. These are my original pictures, actually, for the two different designs of my devices. You can see I have tremendous handwriting. It's super beautiful. I got in trouble with the patent office when I tried to submit that. They said, no, you have to redo this and have a CAD drawing done. And so the question is, do you license it to a commercial entity? Sometimes that can be the best way. And, it also depends on what your particular level of interest is, right? Do you want to develop the device yourself, go through all the work and hardship of device development, which means you make more money on the back end, but it also means that it's hard, right? If you go to Cook with an idea and they decide they like the idea, their terms are terrible. Don't ever work with Cook. But if you were to do that, they'll promise you 2% of whatever the gross of the device is, and that's it. That's all you get. You know what I mean? That's the terms. You get that 2%, eat it, but they'll do all the device development for you themselves. Whereas other companies have much more profitable terms, you know, and again, you have to decide, okay, am I going to develop the idea myself? Am I going to start a company to develop the idea? Is the university going to build the product, get it all ready to go, and then sell the product itself to a company or license it to a product? And are there grants that I can use to support the research? There's NIH grants that are set up specifically for designing devices for underserved areas in medicine. So, and those don't require you to pay any of that money back. So you get an R01, which requires you to work with a company directly to develop a device. That can be a good source of money that you don't have to pay back. Venture capital, you just want to make sure that you're very careful about who you work with, because venture capitalists are always there to steal your money. And not 100%, some of them are really good guys, but and ladies. But the key is you want to make sure that you're very careful about that, and that you have somebody smart help you talking venture capitalists, because basically they're all about the money. They're all about the Benjamins. So you got to make sure that the terms are not totally unfavorable to you and totally favorable to them. And then do you do self-funded research? Some of you say, okay, I only need, say, $50,000 to develop my device. And then if I get it through the FDA, because the thing is, the level to which you develop a device is directly proportional to the amount of money that you get on the back end with the device. So the further you take a device before you engage investors, the more money you're going to make, right? And, you know, so that's always something to consider when you're thinking about this is, okay, how far can I take this device by myself? How much is it going to be worth on the back end? So if a device is going to be worth $100 million, the entire market's $100 million, and it took you $1 million to get it to the spot where, you know, you can get through the FDA, and you do that all yourself before engaging investors, then you're going to make a ton more money on the back end. Whereas if you engage investors when you have no money and they put the $1 million in, then their equity share of the company when it sells for $100 million is significantly higher, and you're going to end up with a much smaller chunk of the money. So you want to be real careful about how all of that is sort of set up. And, you know, in some cases, there are good people to work with and bad people to work with, just like anything else. It's just money is a lot more prevalent in this. And so there's a lot less altruism. So it's something to really sort of be careful of when you're doing this kind of stuff. And the thing is, if you work with the university basically has no vested interest in ripping you off. The terms are see-through at the beginning. And so while their patent lawyers are not as good, 100% not as good, and while their lawyers are not as good, the fact is they do actually have your best interest in mind. So that's kind of the benefit of working it through a university, is that you at least have the benefit that somebody's kind of looking out for your interests. And who can give you research assistance? Do you do all the research yourself? Do you get somebody else to do it? There are labs that will actually do FDA testing for you completely intact, but you know, that's a little expensive sometimes. So you just have to figure out what you have in terms of your resources and how much work you're willing to do. You know what I mean? Some people want to start a company, be the CEO, kind of develop that company, grow the company, and then potentially sell it down the road. But they want to actually develop it like sort of from the idea all the way through the company. Some people just want to have a great idea and have somebody else say, that's a great idea, I'm going to develop that, and you get a little share of the profits. But again, you know, the remuneration at the end is different for those things. And this can be really, really, really big money, just so you know. I don't know if any of you know what this is. Does anybody know what this is? This is the vineyard of Dr. Julio Palmaz out in Sonoma Valley, and those are 3 of his 20 Porsches in his Porsche collection that he has. And do any of you know what Dr. Julio Palmaz invented? The first stent. So Dr. Palmaz, as well as an engineer, were both working at in the University of Texas system, and they developed the first stent. And overall, he ended up making $600 million back in 1991, which is when that stent was bought from him by Johnson & Johnson. And that's the equivalent of $1 billion right now. And so here's the thing. When he went to the University of Texas system and said, hey, I've got an idea for this stent, he went to the technology transfer office, which was even a thing back then. And they said, we don't really think this is that interesting. And so they said, you can, and he said, well, can I have the patent back? And they said, yeah, fine, it's yours. And so he took back the patent and they got nothing. The University of Texas got nothing. And Julio Palmaz got a vineyard and a collection of Porsches. And so he's done all right for himself. And so when we're talking about these device development, it's not money like you see, like the provost of my residency school. She actually is one of the original patent holders for Avastin, and she made $5 billion off of her patent. However, this is not that kind of money, but it's still, we're talking in the billions of dollars with bees, not in the millions of dollars in some cases. And so there's real money here, and that's why industry has been so closely engaged with IR over the years. All right, so I'll go, I'll talk to you a little bit about how I came to my device and what I'm trying to do with my device, which is, ongoing, but that's what I'm doing right now. And basically, I was a grad student / medical student. I did an MD PhD, did my MD at Indiana University, did my PhD at the NIH in the Graduate Partners Program, and knew I was going to be an interventional radiologist when I was doing my PhD, because I actually did the PhD in part because at the time radiologists were making so much money, nobody took them serious as researchers. And so that's not as true anymore, but at the time it was very true. And so I kind of wanted to make sure that I would get taken seriously as a researcher. And I did pathway biochemistry and metabolism. And in fact, my PhD advisor, one of the two of them, because I had one at Indiana and one at the NIH, he's actually the one who discovered branched-chain dehydrogenase kinase. And basically maple syrup urine disease, for any of you who care. But anyway, he also did a lot of pathway biochemistry. And so it was really interesting to me after I started to learn a little more IR. I'm like, it's kind of crazy to me. All the amino acids have secondary signaling functions. And so when you eat, you put food in there, right? Cheetos and Cheese Whiz and burgers and all that stuff go into your gut, which then comes into your portal vein, which goes through your liver, and then all of those amino acids get balanced because they're signaling molecules. They're not just substrate, they're actually signaling molecules. And that's how your liver, kind of the organ of homeostasis, signals to the rest of the body. And so that makes perfect sense. But the problem is, when you look at it, when we give IV nutrition, we don't put it in the portal vein, we put it up here in the hepatic vein on the other side of the portal vein. And so that doesn't make any sense, right? That means that you're kind of going all the way around all of the homeostatic mechanisms of the body. And in fact, if you look at patients who get TPN, they do terrible, and their livers do terrible in particular. If you've been on TPN for 8 or more years, you have cirrhosis. It's just 100% true. If you're on constant TPN for 8 years, if you're a baby, a newborn, like with necrotizing enterocolitis or something along those lines, and you are on TPN for a month, you have cirrhosis, because 85% of all babies who are treated with TPN without oral supplementation for over a month end up with fibrotic liver disease. And so there's even a name for this. It's called parenteral nutrition associated liver disease. And I have a lot of reasons why I understand the mechanism of this better than some, because I'm an interventionalist and know the anatomy, but also know how the metabolism works. I think it has to do with the alanine cycle, but none of you care about that. It's called the Cahill cycle. But anyway, if we were to take the TPN and put it here, then it would probably be exactly the same as if we were feeding people. And then people might gain weight on TPN, and they wouldn't have issues with fatty liver disease and elevated enzymes and loss of growth hormone and hyperglycemia. I think that would be useful. However, we don't have a catheter that goes in the portal vein. The portal vein is functionally different than any other vein. Stable access would allow therapies currently unavailable. The liver creates a specific and unique environment with stable access. And there's a lot of things about the portal vein that are totally different from any other venous system. In fact, all of the blood in the rest of your body is very stably controlled in terms of the osmolarity. The only place in the body that you can have hyperosmolar blood is in the portal vein. The only true venous system in the body that's oxygenated is the portal vein. When you're fasted, your portal vein is where you get 75% of the oxygen that goes to your liver. In a postprandial state, 40% of the total oxygen extraction of the liver is from the portal vein. And 40% of the total lymphatics in the body are on the portal side of the body. So this is a really important and unique area. And, it's the second largest immune complement in your entire body behind the spleen. So this is a really important vascular site, and we don't have catheters there. And, as an interventional radiologist, you'd be like, well, I put stuff in the portal vein all the time. I just don't leave a catheter behind. And so that's kind of, and there are descriptions of doing this in animals because it can be really helpful for doing things like testing first pass metabolism in the liver. And so what they do is they either sew a catheter into the spleen, or they'll actually cut open and then cut down on the left portal vein and then sew a catheter in that way. And this is pretty invasive and not something you want to do on somebody who's super, super sick. So you'd like a percutaneous means. And in fact, people have thought about this for percutaneous access as well. In cows, they actually did it with open cut downs as well. In sheep, they actually do it with big surgical cut downs to put the catheters in. But there was one group in Japan or China, I don't remember, who actually did basically a TIPS method and put in a PICC line through the hepatic vein into the portal vein, although the catheters milked out, so they didn't keep them there. And so my idea was actually to make a catheter that has a balloon on the end of it, because the liver moves all the time and the chest wall doesn't, and you'd rather do this sort of from a percutaneous method. That seemed like a really easy fix to this, and then you'd be in the portal vein. And you think, oh, well, if you have a balloon in the portal vein, aren't you going to occlude the portal vein? Well, no, because I know that from the early literature in portal vein embolization, which we do to try to grow the liver when we're going to do a resection, that you don't cause occlusion of the liver, which is why I knew that you wouldn't cause occlusion if I put a balloon centrally. And so this is kind of what I'm trying to say here, is that, it's the confluence of a specific set of knowledge, because I know the metabolism well because I do pathway biochemistry as a PhD. But then I also know the blood mechanics, and I know the anatomy, and I know the accessibility of the portal vein because I'm an interventional radiologist. And so this is kind of how you find things that other people haven't really done before. And what other uses could we use with a portal vein? Well, it's, obviously my interest initially was related to TPN. But, this, again, as I said, this is the second largest repository of immune cells in the body. It's the most common site of metastatic disease. And, if you're talking about immunotherapy and that sort of things, there's more dendritic cells in the liver than there are anywhere else. And so you have kind of a confluence of issues that potentially help you to be either more efficacious in terms of being able to deliver higher doses of chemo if you could go into the portal vein, which is the side where the metastases go, by the way. So you will hear all the time, these are arterially enhancing, so they get all their blood supply from the artery side. Yes, that's true. But if you're talking about immunotherapy, the immune cells are primarily on the sinusoidal or on the portal venous side. And in addition, the hepatic arteries and the portal veins dump into each other. And so this is a potential area to get immunotherapy or immune-specific treatments that you can't put elsewhere in the body. There's actually a CAR T cell that's currently designed that has specificity to attack cells that express the CRC receptor. So you can't put those in the in the peripheral circulation, because then their colons will rot and fall out of their butts. But if you put it in the liver in a person with liver metastatic disease, you could potentially kill those cancers without affecting the rest of the body if those cells stayed in the liver. And then the other thing about this to know is that there's some interesting newer data that says, okay, in patients who you treat with immunotherapy, you're actually more likely to be successful if they don't have liver metastatic disease than if they do. And, given the frequency with which we metastasize to the liver, this is a very attractive idea for a portal vein catheter. And just so you have an understanding of why people are so jacked up about immunotherapy, this is a patient of mine. I treated her HCC with TACE. It laughed at me. We put her on sorafenib. It laughed at sorafenib. We tried TACE again on sorafenib, laughed even further. And that the picture on the left is her right before we started immunotherapy. She has Mets to her uterus. She has a huge Met that's compressing her airway, that's pushing into her thyroid, and she's basically on death's door. She was an ECOG 3. Her albumin was less than 1, and she was about to die. And so we put her on pembrolizumab, and her alpha fetoprotein went from 160,000 down to 100. And she got 2 years in which she had 0 symptoms from her immunotherapy, and actually got better and turned into a normal member of society. So she got 2 years of solid survival out of immunotherapies. And what she finally died of was actually immune problems related to idiopathic thrombocytopenia purpura, probably caused by the immunotherapy, but because her spleen was getting too much of the immunotherapy and not enough was going to the other parts. And so this is the kind of patient that we feel like might be able to sort of be helped by this kind of technology. Also, consider a lot of the current chemotherapies have significant liver toxicity. So if you had a catheter that was a selective infusion catheter that you could put in the liver, you could protect the liver while, treating the rest of the body. And then also there are specific chemos that we infuse directly into the liver. And you could actually do 5-FU into the liver. It has 96% extraction, which is why they like to do infusion with that. And, currently what they do is they do half a Whipple so that they can expose the gastroduodenal artery, and then they sew a catheter into the gastroduodenal artery, which requires the patient to be continuously anticoagulated. And then they, close them up with a pump so that they can get, occasional 5-FU infusions. But that's like a 4.5-hour surgery to put that catheter in, and they have to be on anticoagulation. Whereas if you had a catheter like this, I can put these catheters in 30 minutes, and I don't have to cut anybody open to do that. And I could still give them exactly the same effect in terms of the 5-FU getting selectively more to the liver than to elsewhere. So there's lots of interesting reasons to get a catheter into the portal vein that I didn't think about when I initially thought of the device. It's only after going, you know what, that would be really cool if we could do that too. But the original idea was actually related to TPN. This was my initial design. It's basically what I did was take a standard double lumen chest port, and I, me and an engineer created a balloon attached to the end of this, and one of the infusion ports filled up the balloon, and the other infusion port was the infusion device. And so we, this is something that's a fact of life. That's the balloon inflated, and it's hard to see, but actually it's squirting out the side because we figured out that different kinds of glue don't work. And, this is kind of what the balloon looks like on the end of the catheter filled with dye. And so the key is, once you get to this point, you have an idea, you're starting to develop it, you have your so-called MVP, minimal viable product. It's kind of like ditch digger school. You just sort of have to keep with it, and it's problem solving because you're doing stuff that nobody's done before. And so this can be very painful and not incredibly straight lined. And so you really just sort of have to keep with it when you get to this point. And so, this is my initial portal vein catheter placements. You can see I'm taking pictures with my iPhone because I'm using garbage equipment in a lab. And, I've got a catheter in the portal vein of a pig. And it was hard, not easy, but, you get there. And then, you kind of move on from there, and it gets a little bit more fancy. And this is sort of what our device looks like now. We have, 2 septums. We have the silicone resealable septum, which is just like a normal chest port. And then the balloon lumen is actually that thing on the side, the plastic thing on the side that you can inflate so that it's impossible for a nurse to inadvertently puncture the wrong membrane and cause trouble. That was kind of part of my design. And one lumen is smaller than the other so that you get the biggest infusion lumen you can. Well, the smaller lumen is you actually fill with air for the port. It looks pretty fancy when you see it in a 3D reconstruction using a much fancier equipment than I originally had. That's a CT scan of this port in a pig after we just placed it. And the beautiful thing is we saw no histologic changes associated with the catheter. And so, basically the roadmap is pretty much the same for everyone. And this is our roadmap, is proof of concept, device design, animal testing of the near final design. And then, you apply to the to companies to get you funding, do all that kind of stuff. You go for what's called 510K approval. In my case, the key is you actually have to ask the FDA, okay, what do you think, what kind of approval am I going to need? And so in some cases, you won't get away with doing a 510K. 510K means that it's close enough to a lot of existing devices that you don't have to do a bunch of in-human testing. And it also depends on how dangerous they consider the device. One of the things that you are really lucky if you can do is you try to design your device so that it is the simplest that it can possibly be, which is what I did, and is made of a bunch of requisite parts that are already approved for other devices and people, because then the FDA is not super worried that you're going to have something that breaks down because they haven't seen it before. And, then you start doing animal studies. And, here's kind of where we are right now. We're actually, I've talked to multiple companies about developing the device. We've created a company. That's what that little Thor logo is up in the corner. And there's basically 3 of us. There's me, an engineer, and the money guy who put the original money in. And basically, we've done proof of concept. We have this. I thought I actually had pictures of my other slides in here. I'll find you another one in a second. I thought I had the other picture of my slides. But basically, we proved that we could put these in animals. We've kept them in for a month. We actually did MRIs on the animals and did histology on the animals at a month. So we've proved that our device design works. And now we're doing some studies to prove that the TPN works. Because here's the thing. I went to these companies and I said, hey, immunotherapy, this is potentially really important in immunotherapy. And if you look at the number of patients with cancer who are candidates for immunotherapy, it's 45%. And if you look at the candidates for immunotherapy who have cancer, that 45%, 75% of those guys have liver mets. And so this is a potential use in that number of patients. And immunotherapy is incredibly expensive. And so you're talking about a market that presently is worth $175 billion a year. And so, you go to the companies and say, doesn't that sound very exciting? And they say, okay, I see that you can get to market really quickly through a 510K process, and that's great. So then you've got FDA approval, but then you don't have a specific indication. So how are you going to get people to buy this? Because you can't market it for anything that you don't have an FDA indication for. So that's a really important thing to look at. And so one of the things you should think about, which is something we had to think about, is that you actually have to figure out what is the fastest way to get an indication for your device that will actually then allow you to sell the device. And then if you can sell the device, what can you sell it for? And what can you charge for it in terms of the procedures that you do? And so this is all a bunch of business stuff that isn't that exciting if all you're thinking about is, oh, I can make this and it will help people. But at the, nobody likes to know how the sausage is made, but if you don't know how the sausage is made, people are either going to feed you bad sausage or you're not going to be successful in what you do. You know what I'm saying? So it's really important to kind of have a good idea. And the best way to do that is to find mentors who have no interest in your device at all. Just don't have any financial interest at all. Unless you engage them to do a significant amount of work for you, then you probably should give them some interest. But you want people who don't have interest in your product to be sort of, objective helpers in doing this. And so I talked to some other people who I know who are pretty good device developers. Ernie Ring, he's got 8, 10, 12 different devices on the market. So I talked to him. And then I talked to some other people I know. There's a guy named Matt Dreyer, who's the one of the directors for research for Boston Scientific. He and I were grad students together at the NIH, so I talked to him. And then for the FDA, my PhD advisor, his wife, happens to be the FDA person who wrote the guidance on device approval in people's, because she's a veterinarian. And so you go to the people in particular people who don't have interest in your product, so they have no vested interest in kind of hosing you, and get lots of advice from stuff like this. But make sure that when you're talking to people that you're kind of protected, because you want to make sure you don't get, jacked by somebody because you didn't appropriately protect yourself. And so in many cases, when you're in the middle of this, before you have a patent, you have to do things like non-disclosure agreements, an NDA. And basically that says, okay, you're telling me about your device. I can't develop the same device because you told me about it and I agreed I wouldn't. So when I went to talk to companies or talk to my friends, I had to make all of them sign NDAs to make sure that I didn't get ripped off. Do you know what I mean? And so there's a lot of those kinds of things that you just really, it's better to know up front. And so the best thing is, if you really have an eye for design, to make friends with other people who are further along than you, who've developed devices, and then ask for their help when you're trying to decide how to get to different places like this. Most of the people are actually very, very sort of friendly about that, is what I would say. They all were for me. And again, Ernie Ring's not getting any money from this catheter, you know what I mean? And so having his input is very helpful because he has multiple catheters on the market. And I wanted to make sure that I left time for you guys to ask questions because I think, you know, this is kind of a confusing subject and it's difficult for me to know exactly what's the most important for you to hear. So I just wanted to make sure that we left time for that. So if anybody has any questions, I'm happy to take them now.
All right. Awesome.
And sorry, I talked a little fast because I wanted to make sure I could get through all of my slides and still leave time for questions.
Okay, so we'll open the floor up to any questions. Just unmute your mic and then ask if you have any.
Hey, Dr. Johnson, can you hear me?
Sorry, I was drinking. Yes, I can hear you.
Okay. I was just going to ask, like, the 5K. I remember I saw like some kind of like YouTube video about that, how you can expedite your process, and you talk about that. So is it two different issues, or is it the same issue that, like, whenever you try to make a similar device, do you have to use, like, I heard you talk about the indications that the FDA would approve it for? Like, if you use 510K, are your indications automatically like given to you based on the fact that you're basing your device on similar devices, or do you still have to apply for different indications versus those similar devices?
So it depends. Probably if I got my device approved from a 510K, it would be for vascular infusion. And then anything beyond that, any indication beyond that, I couldn't market it for unless I actually had that indication. So for instance, you know, and sometimes that's a problem, sometimes it's not. So like if you look at all of the stents that are on the market now, aside from the Palmaz stent, and aside from the Gian Turco stent, and then the brand new stents, most of the stents that we have on the market actually were not approved for vessels at all. That's the only place we use them, but they're all approved as biliary stents because they're all comparing themselves to the wall stent. And so they're getting essentially the same as a wall stent, which means their indication is actually biliary stenting. Now, everybody knows that, and stents have been around long enough that none of us pay any attention to that. So almost every single stent that's used in clinical practice is used off-label. But that also means that those companies can't market them for those other things. So I can't talk about how great, and I can't have, you know, reps come and talk about how great my vascular stent is because I'm not allowed to talk about it unless I have an indication. And so there's limitations in how you can market it more than anything else. But doctors, once it's got FDA approval, you can use it however you want. You know what I mean? Like there's no limitation on you and how you use it. It's just a limitation on marketing. So for my device in particular, we would definitely get a 510K, but then we would specifically go for indications that would then allow us to actually market it to get people to actually use it. And so that's kind of what we would do, is go after TPN first, because that's the quickest and easiest way to get to market with this particular device, because that doesn't take a real long time. I can do and if you want to know the specifics, we're going to do a one-to-one comparison of patients in the ICU getting short-term TPN with my device versus standard TPN, and we'd use standard, like off-the-shelf TPN versus the stuff that the hospital makes. Because what you're trying to do is find a differentiator. And so I could do that in a very short study. Basically, I'd have to pay for the TPN, I'd have to pay for the catheters, but everything else that we would want from a data standpoint, including daily labs, when patients are discharged from the ICU, daily weights, those are all measured anyway, right? And so I don't have to do a lot of work. And then if I have an indication that says this is better for TPN, then it becomes very easy to sell the catheter. Because otherwise people are like, okay, you make a catheter, it sounds great, but why would I buy that instead of just a PICC line or instead of a chessboard? Sounds way more invasive. Why would I do that? And so in some cases, when you're doing something that's really new, you kind of have to have an indication so that you can actually talk to people and get them to understand why it's important to use your device versus somebody else's device. Does that make sense?
Yeah. And if I could ask a follow-up real quick, but so like, because you talked about how a lot of these stents are, they were compared to a biliary stent initially, and that's how they got their 510K. How are there parameters on like how close your intended product needs to match to a previous patent to like get approved by this process?
Well, so it's totally up to the FDA. They get to decide whether they think it's fundamentally the same as an existing device. And they do that based on materials, they do that based on design. And again, wall stents, nobody uses in the bile ducts anymore. They only use them in veins as well. So even the wall stent, which was originally approved as a biliary stent, we all started immediately using it in veins as soon as it was approved. It was just easier to approve from a biliary standpoint. Does that make sense? So we are basically using it exactly the same as a wall stent, but it's a judgment call on the basis of the FDA. So that's why you got to go to the FDA, show them your device and say, hey, what's the mechanism that you think this is going to require? Because they actually have to make an official determination before you submit your application. So you have to, so it's always good to go to them and say, what do you think this is going to be? And so I went to the person who does those determinations and said, hey, what do you think this is going to be? And because I originally thought I was going to try to do something through a humanitarian device exemption, because again, I'm just looking at the way that I can get it into people as quickly as possible with as few people actually doing the research on as possible, because every person you add to your studies, that's a lot more money. And again, like we talked about, TPN is not nearly as big a market as immunotherapy. Won't ever be as big. The entire worldwide market for TPN is about $9 billion, whereas I told you the worldwide market for immunotherapy is $175 billion. So that's two orders of magnitude bigger, right? But the issue is, in order to do the cancer study, that's a longer study because you got to look at survival and you got to look at dose, and you've got to start with a phase 1 and then go into a phase 2. And so it's a much, much harder study that's harder to do when you don't have, $100 million sitting around in your pocket. And so that's kind of why I chose the approach that I did. And that's kind of one of the things you have to think about is, what do I have to do to get it through the FDA? And then once I'm there, what's it good for? Why would anybody use this device instead of the existing devices? Because there's lots of devices out there. And so that's kind of how we approached it. But I didn't know that we'd be a 510K mechanism. It was only after talking to the FDA that I knew that. And so once you start, file the patent, and by the way, filing the patent actually gives you protection until the FDA either accepts your patent or rejects your patent. Oh, and one thing on that, I should have said something about that. That's a really good point. When you actually write your patent application, make sure you talk to somebody experienced and not just the patent lawyer from your university before you submit your application. Somebody who's done patents before. I kind of got hosed because my patent lawyer gave me at Colorado gave me no guidance at all. He said, okay, tell me about your device, submit it, we'll submit the patent. And I said, okay. And I thought, oh, I want it to be as broad as possible because that's the way that it has the most potential uses, right? But that's not how you want to write a patent. You want to write a patent to make it as specific as possible, because when it's too broad, it has parts that are too similar to other patents, and you can't get a patent issued if what you do is too close to something else. And so you need a bunch of differentiators that makes your device completely different than other devices. And so if you don't have that, it's very hard to get patents. And the thing is, if you submit it and it's too general, then the problem is that's prior art. It's called prior art, which means if you go to file, so say I file a patent, it's way too general, it gets completely rejected by the patent office, right? Then I say, okay, got rejected, too non-specific. So now I'm going to make a very specific patent, and I talk about my device exactly, and it's totally patentable in the form that I talk about it there. However, you know what they would use to reject my new patent claim is my old patent claim, because that becomes prior art by itself. So I would have to completely redraw it and find some way to make it different from what I claimed, even though they didn't give me a patent, right? Because any patent application is potentially prior art. And so they had crappy patent lawyers at University of Colorado. They have better patent lawyers when I'm working with people who actually know what they're doing. And so my patent did get issued, but I struggled really hard to get my patent issued because I had a patent lawyer who didn't tell me that I needed to be as specific as possible. Because if I had been more specific at the outset, then I wouldn't have had any trouble, because eventually my patent did get issued, but I had to find all kinds of way to argue that these other 12 devices that they're trying to say are the same as mine aren't the same as mine, and no combination of person would think to put all the pieces together that I'm talking about to make what I'm making. And so it's really, really a good idea. That's probably the most important step if you're doing this. When you're writing your patent, make sure that you talk to somebody who's kind of smart and not just the patent lawyers who work for the university, because it turns out that the ones who work for the university are the ones who are not working for the device companies, who pay way better than the universities. And if you're a patent lawyer, that's what you care about, is how much they pay. And so from that standpoint, it's a really good idea to make sure you're as specific as possible and that your pictures are as specific as possible, because again, it's all about differentiators of other devices. So that's the one piece of advice I would give now that it took me forever to get my patent issued, and I had to issue multiple responses and it was just terrible. But that's the kind of stuff that you want to think about before, when you're writing it the first time, because once you put it down on paper, it's prior art and you're kind of hosed if you don't have it written in a way that it'll help you. That's a little bit of a tangent. I hope that answers your question.
Yeah, and if I could follow up just one more time, I'm curious. So like, let's say that you make your device, you get it to market, and you're marketing it, like you said, for right there in the patent or in the portal vein, I'm sorry. And so let's say that you put it to market, and then people start using it, kind of like how they did with the biliary stents and the wall stents, that you go and start using them for different things that you didn't, that's not your indication, and you can't market it as such, but you want to go and get this new indication. Do you have to redo all like your studies proving that your device works for that indication, or can you use like off-label, like, hey, look at all these people using it as off-label, obviously it works for this indication, can I just go ahead and add this indication onto my device so I can market it?
No, you actually have to do research to prove the indication. And so what a lot of people do is they'll go to Europe through the CE mark process, and they'll do their research in Europe with people who are already collecting the data that they want. The problem is you need prospective data. The FDA won't accept anything but prospective data, and the FDA is the hardest to get through relative to other approval authorities. And so you really what you want to do is do a prospective study. And so yes, the reason that nobody has done prospective studies to prove that the wall stent is good in blood vessels, or that any of the other stents are good in blood vessels, is that the pathway to do that is just expensive, right? You'd have to do a prospective study. You'd have to tell patients that it's experimental. You probably wouldn't be able to charge for the stent if you're doing research on it. So you run the risk of losing a whole bunch of money, but you would have to do it prospective. And basically what everybody has done in terms of the wall stent in particular is all the other companies that came out with stents, instead of marketing it for blood vessels, what they say is, hey, it'll do the same things a wall stent does. And we all know what that means, right? They don't have to say, oh, it's good in the bile ducts. We will know that it's good in the blood vessels because we all know what the wall stent does. And so the way that these companies market them is they say, hey, this is functionally the same as the wall stent, but here's the little things that make it better. And so they're not talking about specific indications. And in the case of stents, it doesn't really matter. In my case, it probably would. And so not only do you get the benefit of being able to get new indications by testing new indications in these cases, but sometimes you can also then do a use patent, which is another thing that can be very helpful. Like, not only did I patent that this is a catheter with a balloon on the end, blah, 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 but I also patented it as a device that is used for portal venous infusion. And since there's nothing else on the market that does that, I win. and it makes it very hard to develop a competitor product to me because I already patented something for use, which means even if somebody else developed a catheter that could be used in the portal vein, they're not allowed to market it that way. They can cite studies that did do that, but they can't actually market it for portal vein infusion if I have a use patent saying mine is the device for portal vein infusion. And so there's a lot of little subtleties to the whole regulatory environment in terms of being able to, what you can market and what you can't. So it's a little tricky sometimes. And also in terms of reimbursement, sometimes insurance companies use approval status to decide what they'll reimburse and what they won't. And so that's another thing you have to be worried about when you're doing this. This is all very, very high level, but it is stuff that you want to think about at one point or another. But to answer your question, no, they would not just give you an indication because everybody's using it that way. You'd have to do a prospective study to prove it. And it has to meet FDA requirements. There's specific requirements for an FDA compliant study. That's the other thing, is that like if I'm doing animals data, which is what I'd have to do to get my 510K, I'd have to prove in animals that it worked okay. And they would tell me the number of animals I need to do, but I also have to do them in a way that meets all of the FDA controls. So they have a bunch of things that you have to do in the study in order for it to be an FDA compliant study. So not only do you have to do a new study, but you have to do it in a manner that's FDA compliant. So it's a pain. And so if companies don't think it will affect their ability to sell their product, they won't do it. But then you run the risk. We're actually engaged in a trial right now with a product called Lava, which is a kind of glue. It's like super glue, but not. It's exactly the same. The product is exactly the same as another glue called Onyx, which has an indication for gluing aneurysms in the brain. But Onyx is used all the time in the periphery, but they never got an indication for the periphery. So Lava is now doing research to try to get an indication in the periphery so they can steal Onyx's lunch. And because Onyx never did the work to actually get approval for peripheral embolization. And so this is the kind of risk you play. You know that people will use it, and you can say, oh, look at these studies of that other people, and it's amazing how careful you have to be with your words sometimes when you're talking about this. But you know, reps can come up to you and say, hey, look at this new study showing what Onyx can be used for, and they can show you that, and then you see data from other doctors, which is more compelling than some study done by a company anyway. But then also they, you know, that's the only way they can talk about it. But then if somebody else gets an actual indication, they kind of get screwed on the process. So this is kind of, it's a weird game that you have to play. And some people like it and some people don't. And if you don't like it, then probably you just want somebody else to develop your idea and send you a check. But if you do like it, or if you're willing to sort of navigate those, it can be very fulfilling things to do. And in particular for IR, our world changes every 10 years. Like what we do, the complete list of what we do as doctors completely changes every 10 or 12 years because new devices come out that just change everything. You know, if you talked to me 10 years ago, I would say we were doing mostly RFA, right? Radio frequency ablation for our ablations. Now we're doing mostly microwave. And it's because the device that we use that's a third generation microwave that's actually good and much, much better than RFA, it didn't get FDA approval until 2013. So the device that everybody's using has only been around for that long. You know what I mean? For ablation. So, and we're much more dependent, like people still do cholecystectomies basically the same way they've done them for the last 20 years. Once they invented cameras and they were doing them laparoscopic, they did them the same. And if you do an open cholecystectomy, you're doing it the same way that you know, what's his face? William, gosh, forgot his name. Oh, that's terrible. I'm such a bad person. William Halstead did on his mother on his kitchen table at 2 in the morning, the very first time a cholecystectomy got done back in, you know, 1898. You're still doing it basically the same way that he did, using similar devices, but IR, none of that is true. Everything is different. And so you have to sort of be a person who's comfortable with your life changing a lot as you go along. You know, when I was in med school, everybody was doing vascular disease all the time. Now we're all doing oncology all the time. And I do a ton of pain. So it's a rapidly shifting world, and you just have to be okay with that, and you have to figure out how to navigate it based on what you're doing.
Then one final question. So you said that like Lava can come and they'll try to like steal Onyx's lunch because they can get this indication for approval. So like obviously they can market it because they have the indication, but when you say steal their lunch, do you mean by just the fact that like how insurance compensates, does that also change based on the indications for devices versus just people using them off label?
Sometimes insurance will control how you can use a device in terms of what they'll allow. In this case, probably what will happen is because Onyx and Lava are exactly the same thing. It's the same kind of glue. They both have tantalum in them. They're a little tiny bit different in terms of their delivery system, but it's the exact same thing. What they'll do is they'll just be able to market it as a peripheral glue, and they'll also be able to go to all the doctors and say, hey, this is a peripheral glue. I know you may have tried Onyx in the back, but really that's just for aneurysms in the brain. We actually have an indication for this peripherally. So it gives you a differentiator so you can go to people and say, look, this is what you should be doing and not what you're doing now. So. But yes, in some cases, insurance companies won't let you use the device because they consider it experimental. That's something insurance companies love to do, is declare things as an experimental treatment. And if they do that, then you're not going to get paid for it. So if you have a specific indication, and in particular, this is a little off topic, but if there's a cancer treatment that actually makes its way into the NCCN guidelines, then insurance companies have to pay for it. And so they don't have a choice. That's the rule for the government. And so depending on how you set yourself up, you can make it so that people get reimbursed using your device and not somebody else's device.
Okay, do we have any more questions currently? Those are great questions, by the way, Nathan. That was pretty insightful. All right, I think that's all the questions we have, Dr. Johnson. I appreciate you giving the time to give a lecture. That was really actually very interesting and informed a lot about device development. I think a lot of us here, speaking on behalf of them, just have no idea really how it works. So that was a really nice insight for that.
Well, hopefully it's helpful for those of you that have interest. And again, you can always email me and ask me questions if you're developing some device. Just make sure you've disclosed. I'm not going to steal your device. I don't care, but make sure you've actually done your invention disclosure first, because you want to do that. That's kind of the first thing to do. I don't think any of you as students actually count as being subject to the guidelines for the university. In some places, residents are, in some places they're not. So that's another thing to think about. But once you're sort of out in the world, just make sure you get advice from people who you trust and people who will help you, who have sort of been through the process before.
Absolutely. Well, on that note, we will go ahead and sign off. I appreciate everyone coming, and especially Dr. Johnson for taking the time to give us a lecture. And if you have any questions, you can email us or you can email Dr. Johnson specifically. Hope everyone has a good evening.
Yep. Good night.
Thank you.
Thank you.
Thank you.